at 6,000 RPM, which, if you've ever looked at a centrifuge, that's incredibly fast. So this little mechanism is going pretty fast. Now, the energy of how these protons are coming through here and running this mechanical device is what we call this proton motive force, and it really has two parts to it. It has the change in pH, that is, the pH is much lower over here because there's more protons. And so when you talk about a concentration difference, as in the case of pH, across that membrane, inner membrane, you're talking about entropy, delta S. So in the equation, delta G equals delta H minus T delta S. T delta S is a form of energy, whether you like it or not, whether I like it or not. The other part is, by pumping a proton across, you create this negative charge on this side. And of course, the protons would love to thermodynamically be attracted towards that negative side. So that's this portion over here. The change in potential, delta psi, psi. So delta psi is a contributing factor. Does that make sense? So both of these humans call the proton motive force. So the tendency of that proton to want to come rushing through this rotor from this side to over here depends on two things. What's the pH difference across that membrane? And what's the charge difference across the membrane? And these get complicated because there are some other systems that run off proton gradients and that generate charge differences too. So looking at it just in this window with blinders on, you're not seeing the full picture. Is that okay? So this looks a whole lot different than glycolysis and making GTP in the Krebs cycle. This is not, I can follow high energy phosphates. This is all about protons. So as you can imagine, this idea was very hard to accept for biochemists for a very long time. Dr. Mitchell is the person who came up with what he called the chemiosmotic theory, and for this he received a Nobel Prize. But I tell you, this was 20 or more years of arguing in the literature with some really weird theories, and very slowly the experiment started discarding these bad theories. What was thought to be a crackpot idea turned out to be the way life does it. So, pretty neat. ATP synthase is this guy. The Greek letters, these alphas, this is an alpha, this is a beta, and there are three pairs of these. So there's one alpha-beta pair. Back here is another alpha beta. Here's another alpha beta. All the alphas have the same sequence. The betas all have their identical sequences, but different from alphas. Gamma here is the crooked finger that's reaching up into here. So you can think of alphas and betas as being kind of like slices of orange. And the orange is held in place by delta and these B subunits. There's two of them. Whenever you see a weird structure, you think, well, what's its function? And so here's one B, and the other, its brother, is right behind it. And these two are making contact up here, but they're embedded in the membrane. And so this pair of B's are coming up here, and with this delta are holding on to the top of the orange, so the orange doesn't just fly off. So what's moving? Well, first of all, FO is A, B, and C. A is this part here, which they still don't have X-ray data for its structure. This is a protein that's embedded in the membrane, and things that are embedded in membranes, which are integral proteins, are hard to get crystals for. B we've just talked about, that's the support thing that comes up here. And C are all of these subunits that make up this rotor. So here, this diagram is showing it for yeast mitochondria. And we'll focus our attention on the yeast cells. Okay? Yes? Yeah, in other words, what's moving here are these C's. So let's talk about these C's. Let's pretend we're talking about the yeast. There's 10 of these C subunits. So right here is one subunit that comes all the way down to here. And then there's 10 of these in a row, identical. And in the center here, we think this open area is filled with lipids, which is embedded in the membrane. And so these 10 guys are turning around like this. And when they turn, this gamma, which is the crooked finger, is attached via epsilon to this base plate. And so when the bottom ring of 10 C's turns, it's turning the attached stalk, which is the gamma. And that gamma is going up inside here and kicking each alpha-beta pair, one at a time. So, it turns out, as you might expect, that in other organisms, as explained in C8 and C15, the base plate, the rotor, down here, has a different design because the proton motive force isn't exactly the same. In other words, remember the proton motive force is how many protons, what's the pH difference across that membrane. That's not going to always be the same for every organism. And secondly, the net charge across that membrane won't always be the same. And so these things are tailored so that some organisms, like E. coli, have a different number of C's in the rotor compared to you and I. So for example, you and I only have 8 subunits down there instead of 10. So it only takes 8 protons to move the rotor one turn. Whereas in the yeast, it takes 10. And in some other organisms, it takes 15 protons. So in other words, the energy per proton is never going to be exactly the same in every organism. So let's focus on the 10, okay? The yeast. That's where most of the work is being done. So you've got this assembly here, the B's, coming up here to make sure that these guys, the alphas and betas, don't just fly off because of this internal stalk, gamma, that's rotating. A here, along with C, when the C comes and touches right up next to the A, there are 2 half channels. One half channel allows protons to come in. The other half channel on the other side here, they're very close in space. This diagram makes it seem like they're far apart, but they're actually really close. A proton can come in. It goes for a ride on the rotor, on the C subunit, and then goes out. So what I like to think of is to think of the protons coming in down here as somebody with a hose that turns on the water or the proton. The only way to exit is going around the rotor and squirting out. And it's the force of that water that's making that rotor spin at 6,000 RPM. Cool? Who would have dreamt this up? And right here in this, they're showing that the catalysis is occurring right at the crevice here of each alpha-beta pair. So the crevice between this alpha and beta pair is where the enzyme active site is. It binds ADP and inorganic phosphate and makes ATP. And that's occurring at all three binding sites. So this ATP synthase has three enzymatic sites at each one of the crevices between the alphas and betas. Cool? Again, that first video thing on homework 3 shows more than what you want to know about what's happening here. Okay, I'm just asking you to understand the concept. We're still going to tear it apart a little bit. Go ahead. Yes, and so to better answer that, we're headed that way. Okay. Again, this is all, hi, how are you, ATP synthase, and then I can't believe you're doing this. Because this is not like glycolysis. Okay. So the X-ray structure of this was done by John Walker. And now here, imagine you're looking down, up here on top of where your eyeball is, you're looking down on top of this orange slices. Okay, that's our next slide. So here, this is what people call, the model is rotational catalysis. Everything seems to support it. I mean, technically it's a model, but it's for real. This is proposed by Paul Boyer. And so we're looking down. So here are each of, let's just start at the top here. Here are each of these dimers. And the green arrow here is the crooked stalk, gamma, my finger. You're looking down on top of it, and it's pointing. So we see that there are three binding sites because there's three alpha-beta pairs. And this apparently works in a sequence. So here, this is empty. The alpha and beta sites, again, the catalysis is occurring at the crevice in between. But this site here is now empty. Why? Because it just lost an ATP. Over here, an ATP is firmly bound. And over here, ADP and inorganic phosphate have just come in. So the arrow, the crooked finger here, is pointing this way because it's been spun. And as it goes around, it just kicked these two guys, this alpha-beta pair, sterically kicked them in the pants and made them release that ATP. That ATP is not leaving unless somebody gives it a kick in the pants. 3 protons, H+, remember subscript P means it's starting from the positive side of the membrane because there's all those protons. 3 protons in the yeast model are going through to the negative side, and that's creating the energy for this arrow here to now move. That is, this crooked thing is moving to the next dimer and kicking that ATP off. You follow me? Meanwhile, this one could bind, since it was empty, it can bind ADP and inorganic phosphate. And then it happens one more time. 3 more protons come through. Of course, the protons coming through is a steady stream. They're hopping through. Because each of these half channels, that we're going to get to in a minute, each half channel has got R groups of the protein that want to bind protons to them, plus waters, just like the 2 paths in complex 4. The proton coming in isn't the same proton that actually jumps onto the C subunit of the spinning rotor. You follow me? It's complicated. So the point is the finger is clicking. It's going up the top. I'm tired of going back here like this. I'm facing you like this. The crooked finger clicks 120 degrees. Because 120 times 3 is 360. Clicks 120 degrees. Every time it clicks, it's making contact with the next alpha-beta pair, kicking that ATP off, etc. the beautiful thing about this enzyme, in my opinion, is the way it works is, if you recall from BIS 102, if you had me with BIS 102, Linus Pauling made the suggestion that enzymes have evolved to best bind to the transition state. That was a huge insight. So enzymes, and this is no different, enzymes make the most weak bonds. Which weak bonds? Hydrophobic effect, hydrogen bonds, ionic bonds, van der Waals. They make the most bonds to the transition state. And that's what happens. That's why an enzyme catalyzes a reaction. When the enzyme binds to something, I'm the enzyme. Here comes ADP, an inorganic phosphate. When it comes and binds, I make so many weak bonds to it, I'm lowering the transition state. But what this thing does is it takes Pauling's idea to the nth degree, and that is making the ATP costs virtually no energy, because the enzyme has perfectly evolved to bind the transition state so that when phosphate comes in and ADP comes in, it makes ATP with no energy required. The transition state has gone from very high to 0. That's very cool. So that means it costs the cell virtually no energy to make ATP. But you can't get something for free. The problem then is the ATP is bound very tightly. How does it come off? Oh, the energy coming from the proton gradient is causing the finger to come around and whack it off. Who would have guessed? So the mechanical energy is being supplied by the energy of the proton motive force. Pretty cool. So that's what's happening. So let me just finish this. Walker and Boyer received the Nobel Prize for coming up with this model to explain it. Yes, question? Well, it's making the ATP because without the proton motive force, there's no energy to release the ATP. So in that sense, it's indirectly making the ATP. Otherwise, you'd have 3 binding sites. Well, in this top model here, ATP would not be coming off. Hence, these things would just be gummed up and not doing anything. Cool? So let me just finish one thought here. So the point is, there's nothing magical about this. It's just that this enzyme makes so many bonds to the transition state, it makes the formation of ATP easy. The hard part is to release the ATP. Question?
Why are there different alpha and beta signals? How does it shape
In this diagram here, it's to show that conformationally, they change the conformation depending on how close that crooked finger is coming. In other words, it's not just the tip of the finger. The whole thing is, they're all associated with each other. So here, Lehninger's decided to use this designation to indicate, wow, the conformation is really different than over here or over here. That's what caused that? The finger come along and kick it. So there's 3 different conformations indicating there's 3 possible binding positions. Cool. Let me go ahead a little bit, okay? Because I'm sure I'll generate more questions. So here is an excellent experiment. I still can't believe these people undertook this experiment. Very complicated. So what they did was they engineered this complex here, the alphas and betas, to have a bunch of histidines at this position in the protein. And it's well known that histidines in their base form can form a complex with metals, and they chose nickel. So nickel would be Ni+2. You've got these nickel ions coating this glass slide. And so these guys are going to bind with this orientation, but now they can't come off the slide. Are you with me on that? So they genetically engineered the alphas and betas so that they would stick in this position. Now they've got this thing like this, and now what they do is they engineer the C subunits to have biotin. You know the molecule biotin. And what's different, well, the whole thing is different. They've taken this protein here, actin. Now actin is normally a long protein that's involved in muscle movement with myosin. Actin-myosin interaction. Well, instead they're using it as a tool. And so they did several things to this actin filament. The actin is made up of all these subunits that polymerize and form this long thing. They want it to be long. And in so doing, they make it fluorescent. They put a tag on it so that it glows under UV light. And at one end, they attach a protein, covalently, avidin. Avidin is a small protein, but the trick is when avidin binds to biotin, it's the strongest weak bond known in biochemistry. So if you ever want two things to be glued to each other, use biotin and actin. And that trick has been used a lot in molecular biology and biochemistry. So basically what they've done is they've incubated the actin filament with avidin with this modified C rotor here, biotin, and so the thing would only bind to the biotin. So what they're hoping is, if they can make the rotor spin, then under UV light, you'll see this long filament moving, because it'll glow. Now, I'm not sure you and I would have done all this. This would take years to do this, okay? But then the other major thing that I don't think you and I would have come up with is the fact, wait, we just learned that the rotor runs via a proton gradient. Well, there's no proton gradient. So their thought was, some others had on some other experiments the same thought, and that is, instead of synthesizing ATP, let's put in ATP and go backwards, because remember, the enzyme doesn't pair. Any enzyme will catalyze a reaction left to right or right to left. And so here, the energy of ATP is being used to drive the spinning finger, the crooked finger, which is attached to the plate, and so you're running the motor backwards. Whoa. So, you've got this slide. You put in, it's all in buffer, you know, put in the ATP. You've got a fluorescent light looking at this thing. And what you see is this image. Every 133 milliseconds, you've got to have a way of slowing this down. And so what you're seeing here is this long filament. This is what's glowing. This is the fluorescent actin. And every once in a while, it seems to be going, you know, straight. Oh, and then it clicks. And so it's moved about 120 degrees. And then it goes for a while, and then it clicks again, and then again. So the point is, you can see with this experiment that it really seems to be clicking 120 degrees, 120 degrees, 120 degrees. Only incredibly fast. So this is an experiment to say that this rotor thing is not a fairy tale. People have done other experiments along these lines too. So the rotor is moving the crooked finger, which is sterically kicking the alpha beta subunits, releasing ATP. Now let's talk about how the protons are driving this wheel. here Leninger has this half channel in the A subunit. So the A subunit, along with the C's behind it, are making a channel in the membrane. So a proton comes in and will bind, and I wish they had changed this, but right here, forget about this arginine for a second. The arginine is over here. Right here, what's the proton binding to? This thing is binding to an aspartate on the C subunit. So if we were to take one of these C subunits, so this is one C subunit. Right here in the middle is an aspartate with a negative charge. And this whole thing, the C subunit, really is one alpha helix, but it has two parts. And so right here is where this aspartic acid is, and when a proton comes in and binds, it causes a huge rotation of one helix to the other. I mention this just to say that it's complicated. But the point is, this aspartic acid comes in, and now it's going to carry the proton around the merry-go-round, the rotor. Are you with me on that? And the only way the proton can leave is when it goes all the way around, it hits this exit channel, and now it can be released to the other side. Now, in this diagram, this arginine here, they say that it helps promote the proton binding and helping it to release. I would state it a different way. Here is the half channel where the proton is going to leave. Here is the half channel coming in, where the proton comes in. And here is this arginine right in the middle. So the proton goes on to this C subunit, and then when it comes around, the C subunit leaves. We have a negative charge right here. When this thing loses a proton, can you have a charge in an organic membrane? No, that's energetically not allowed. So in this brief moment, as this thing is spinning, this arginine forms a plus-minus bond, allowing this negative charge to be there, so that when it comes over here, it can pick up the proton. So one thing that the arginine is doing is allowing this for that brief moment to exist as a negative charge on the aspartic acid, so that in this moment, thermodynamics is not violated before it picks up a proton. That's item one. Item two, these are both channels very close to each other. This arginine with its giant positive charge says, proton, when you come in, you're not taking the shortcut. This is not allowed. So if you want a positive charge to be there like a gatekeeper, arginine's your guy. He's going to be positive at any pH. So I would suggest those two things are far more important than stated in the slide. So the protons are coming in and being carried by the C subunit on its trip around and out. Why does it want to go there? Entropy and charge. The charge on the other side of the membrane is attracting that proton to it. Follow it. How this came about, who knows? Now the other thing is, so here in this slide, these protons are going through these half channels. So I wouldn't draw this. It's a straight line going right up through here. It should be going part way in and part way out. But the point is, if this thing is making ATP, so let's focus on this ATP out here. The ATP has to get out to the rest of the cell. The ATP is made on the matrix side. how does the ATP get out to where the cell can use this ATP? There has to be a transport protein. So that's this guy here, adenine nucleotide translocase. Yes, I would like you to know this one, okay? This protein is very specific. It will let one ATP out only if one ADP crosses at the same time going in the opposite direction. So this is an antiporter. One guy goes out, one guy comes in. Just like the transport of carnitine across the membrane in the mitochondria. So what you see here then is something with a -3 charge is coming in, but something with a -4 charge is coming out. So this transport is being driven by the positive charges out here. So in other words, the -4 wants to come out. So the energy of the proton gradient is running this transport. Cool? Getting the ATP out. Remember, it's in a small pool size. It's got to get out to be used. But if we just focus on these guys right here, this guy's got 3 phosphates on him, ATP. This one only has 2. So that would create a problem, and that's where this other protein comes in. Because the problem would be what? If you'd run out of phosphate, how can you make ATP up from thin air? If 3 phosphates go out, but only 2 come in, within an instant you're going to say, wait, I can't make any more ATP. So phosphate, inorganic phosphate, they've chosen this species, but how many charges depends on the pH out there, and that will vary from different organisms. This protein here is a symporter, phosphate translocase. I would like you to know that. The proton is necessary to go in at the same time as the phosphate, so this proton gradient is causing this to bring phosphate in. So from this diagram, every time you make an ATP, you need 3 protons of yeast to run the rotor 1 click of 120 degrees, but it also costs you 1 proton to bring in the phosphate. So that's going to cost you 4 protons to make 1 ATP. And now you see how the ATP is getting out of the mitochondria and how used ATP in the form of ADP and inorganic phosphate comes back in to get made. And so our last slide just looks at the stoichiometry. 3 protons to make that crooked finger go click 120 degrees. 1 proton for the phosphate. It's going to take you 4 protons. 1 NADH causes 10 protons to get pumped across that membrane, according to our first slide today. Complexes 1, 3, and 4 collectively make 10 protons. And so now we can put this together and come up with 2.5 ATP. You can do the same calculation for an FADH arising from succinate. Succinate, when it binds, misses complex 1. So instead of 10 protons, you get 6 protons. See you next time.